Okay, hi, hello everyone. Thank you, Christian, for the kind presentation. Yeah, my name is Andrew Nigoro, so I think the pronunciation was quite good, so thank you. And the title of this talk will be Practical Experience with F3 Searching. So the agenda for this talk will cover a brief introduction to F3s, which Christian will provide. And then I'm going to talk about a retrospective virtual screening exercise that I have carried out by using FTrees, and a follow-up study where I have applied FTrees Farm. And finally, I'm also going to present a prospective virtual screening application.
Okay.
So, Christian?
Andrew asked me to give the part of the presentation about the background of the technology, as this is actually provided by Biosolvity. So the feature trees, very, very briefly, you see them illustrated here, 2 molecules and the 2 related feature trees on top of these molecules. Here in the animation, you see how a feature tree node represents a substructure on the molecule, and the nodes are connected with edges. So the nodes and edges together, they form a feature tree. And you see that these nodes here of the feature trees have colors, and the meaning of the colors is quite obvious. It is the pharmacophore profile of the underlying substructure that is represented. So, for example, the ring on the left-hand side, it has a donor and an acceptor, and it's aromatic in nature, has a certain volume, and the indicator that there is a ring. So a numerical profile essentially capturing the pharmacophore properties. Note that no directionality or 3D, not even 2D information is captured here. It's just the properties and through the connections of the nodes where they are, roughly speaking, on the structure. So this is the descriptor called feature tree. The feature tree algorithm then calculates a mapping of the nodes of one tree representing one molecule and the tree on the other hand side representing the other molecule, the one that you want to compare to. I drew here lines indicating a mapping of nodes from here to here and so on and so far. It's a little bit like sequence alignment, except that these sequences here have branches too. But otherwise, the methodology is really very similar. A mapping will map a node from here to a node over there, and then we calculate a so-called local similarity, which is quite high here for these 2 aromatic rings, and lower for other local matches. Now, among all the possible matches that can be thought of, feature trees finds the one with the highest overall similarity value, and that is the value that you get as a feature tree similarity. So as a user, if you compare these 2 molecules, all of what I said to you happens behind the scenes. The molecule is represented with this descriptor, the mapping is calculated, and from all the mappings, the best similarity value, namely 0.81, is reported back to you as a user. And as an additional benefit, you get to see the mapping also actually through these color-coded depictions, where the same color indicates that these 2 substructures are mapped in the calculation of this similarity value. And here you can see one unique feature of feature trees, which is its ability to ignore the exact substructure and hop to a different substructure on the other molecules. For example, these light green structures here mapped onto each other indicate that it is feature tree found it most similar in the mapping to map this ring to an open chain portion on the other molecule. It highlights one of the key characteristics of feature trees, which is its scaffold hopping capability. And with that, I stop and hand over to Andrew again.
Thank you, Christian. So in the first part of this work, I'm going to talk about retrospective virtual screening. And the aim of this part, let's say, of this study was to assess the ability of FTrees similarity to prioritize non-actives in a retrospective virtual screening setting. And also to compare the results that I could obtain by applying FTrees to those obtained with more traditional 2D fingerprints. And finally, I also wanted to get a feeling of how including pharmacophoric constraints by means of FTrees Pharm could modulate or change the results that obtained with FTrees. In this retrospective virtual screening, I have chosen a virtual screening benchmark set that is called Decoys 2.0, which includes a set of actives and decoys for 81 targets. So for each of these targets, there are available 40 active compounds that are extracted from the binding DB and 1,200 decoys that are extracted from zinc in such a way that they would match physicochemical properties of the known actives. And as ways to measure similarity, I have chosen 3 different 2D fingerprints, which are the max structural keys, ECFP4, so circular topological fingerprint, and GPI DAF3, which is a pharmacophore-based fingerprint, and finally also FTrees similarity. And as a way to evaluate the performance of these methods on the selected dataset, I have chosen to use the area under the ROC curve, basically. And as an example, you can see in the figure, so in the third panel, a ROC curve that in this case, so the blue curve will have an area under the curve of about 0.8, so 0.77, and you can compare this with, let's say, an ideal random selection, which is the black curve that gives an area under the curve of 0.5. So in this case, we have, let's say, a nice enrichment of non-actives, which gives a high area under the curve. Okay, so this is the computational protocol that I have applied, which consists of an iterative procedure in which at each iteration a reference compound is chosen, and this is used to assess the similarity of the rest of the data set, so including both actives and decoys, which are ranked according to the calculated similarity value by using each of the 4 methods that I have mentioned in the previous slide. And the rankings obtained are assessed by calculating the area under the curve, and the next iteration, so the next reference compound is chosen, and the whole process is repeated again. So this is done for each of the actives available for each of the targets available in the dataset. A few virtual screening were excluded because I have obtained, let's say, very high area under curve, so higher than 0.8, with each of the 4 methods chosen. And so I have considered those specific runs as maybe too easy, so not really informative, and I have just excluded them. And this has led to an ensemble of about 2,500 virtual screening runs on 79 unique targets. In this slide, we can see a brief summary of the results that I have obtained by applying these methods. And so in the box plots, you can see the distribution of the area under the curve values I have obtained with each of the 4, let's say, all the 3 fingerprints and FTrees. And I think that the key message here is that the performance, say, obtained with the F-trees is somewhat comparable in terms of median value to those obtained with the GPI DAF3 and DCFP4, while it's superior to the median obtained with MAX. And the median values are also shown in table below. But an interesting feature that I have noticed was that the interquartile range in the box plot for F3 seemed to be shorter. And I have also calculated the standard deviation for these values, and I have seen that for F3s I have the lowest standard deviation. And I think this is interesting because it suggests that the results of the individual virtual screening runs tend to vary less depending on the specific reference chosen for that specific run. And this is probably due to the more or less abstract or generalization of the representation of the structure provided by F3s, which is an interesting feature in virtual screening if you think that sometimes you may have only a few compounds available, maybe you have many and you don't know which one is better to choose, and this is kind of giving you the idea that the results that you obtain maybe will not be depending so strongly as with other fingerprints in terms of the specific reference that is chosen. Christian?
Yeah, I step in again and explain a new feature which we only recently introduced into the feature trees method, and that is Andrew calls them feature trees farm. We call them favorites. So the idea again goes back to this ability of the feature tree software to calculate a mapping between the nodes on the left-hand side to the nodes on the other hand side. And I explained to you how we obtain also local similarity values for these individual matches of nodes. So now, as a user, with this new feature, you can nominate favorites, and a favorite assigned by the user gets a minimum similarity value assigned. So you can say as a user, this part of the molecule is for me more important than the rest because it's for some reason a key component or whatever external knowledge you have, as you usually have with pharmacophore type information. You can use, and you could declare, okay, this must be matched with a minimum similarity of 0.9. Then you already see in this mapping 0.9 is okay because the local similarity is higher than the similarity value. Whereas if you would decide that this is a key feature, it should be matched at a minimum with a local similarity of 0.9, then this would not be valid because here the local similarity value is 0.85. Now, this does not imply necessarily that the whole molecule is now rejected. It only means that this particular mapping between the molecules is rejected. And I told you, feature trees calculates many alternative mappings, and among all the mappings with the favorite feature assigned, only the mappings compliant with the favorite constraints are considered as valid. And as before, among all the valid mappings, feature trees finds the one with the highest global similarity value. So the method in principle stays the same, except that as a user, you have now an influence. You can assign certain nodes here or substructures represented by these nodes as favorites, assign a minimum similarity, and thereby enforce that only mappings where the minimum similarity is obeyed are considered valid, and therefore those molecules are considered valid virtual hits. That's the new feature trees farm or feature trees favorites feature. I should explain how you use FeatureTrees Farm only very briefly. We wrapped this functionality in 9-node. So let's assume you sketch your molecule, your query molecule here. You pipe it into the FeatureTrees query generator. If you execute that node, a simple GUI pops open. You click on atoms, respectively substructures represented by individual feature tree nodes. You assign between 0.8 and 1.0 a similarity value, and thereby you have assigned the favorites. And again, as I showed you before, in the visual results, you can then actually check how the resulting mapping obeys your constraint given through this graphical interface. I want to quickly say a few words about tests that we did with this new favorite feature. We searched for this kinase inhibitor here in EvoSpace, and found among the top 100 most similar compounds, 65% compounds being dockable in the active site of this of the PDB structure behind this compound. But among the hits, we found also a lot of compounds without a proper hinge binding motif, because FeatureTrees doesn't know where the hinge binding motif is. It just takes every portion of the molecule and treats it similarly. With the favorites feature here, this nitrogen and the attached ring set to a minimum similarity of 0.7. At this donor here with a minimum similarity of 0.9, we found a significant higher fraction of dockable results and also many aminothiazoles, so proper hinge binding motifs in the compounds. And last but not least, I want to mention that since a lot of people have asked us whether with this search method they could also do substructure searching, while this is not the typical thing you will do with feature tree searching, with the favorites feature, you can set the minimum similarity of the substructure of interest to 1.0. Then you find molecules only that have a perfect pharmacophore match. Then on the basis of these topmost hit molecules, you can filter by substructure. That's then only a small set of molecules, and essentially this way you have a workaround for searching for a particular substructure also in a chemical space. So that again completes my part of the presentation, this new FTrees Farm feature, and now Andrew can continue with it.
Thank you. So, as a follow-up to the retrospective virtual screening that I have talked about earlier, I have conducted this study by including the FeatureTrees Farm as a development that Christian has just explained, with the aim to get a feeling of whether including these pharmacophore constraints could modulate, hopefully improve the results in the retrospective virtual screening in some specific study cases in which FTrees alone performed poorly. And for conducting this study, I have switched performance evaluation metric from area under the curve to enrichment factor. I have done this because by including these constraints, several compounds tend to be excluded basically from the output of your similarity search, and this will not allow me to calculate a meaningful area under the queue for the whole database, and also this will vary a lot depending on the specific reference and constraints that are chosen. And to cope, let's say, with this problem, I have switched to enrichment factor calculated at 1 and 5% of the ranked database, which basically is telling us how many folds more non-actives are found at that specific stage of the ranked database with respect to an ideal random selection. So to run, let's say, this follow-up study, I have selected 2 study cases. So one target is 11-beta hydroxysterol dehydrogenase 1, and the other target is androgen receptor. And you can see here also the structure of the reference compound that were used to run these calculations, and those were chosen because I could find a co-crystallized structure in the protein data bank for these specific compounds that also gave a poor enrichment of non-actives in the previous retrospective virtual screening, and in particular in area under the queue, which is lower than 0.5, which basically is below random. It's like a deselection of active compounds. So I want to see if maybe modulating the feature tree query by including these pharmacophore constraints, I could recover the ability to prioritize non-actives. So this is the first study case. So for 11-beta hydroxysterol dehydrogenase 1, and in the right-hand side of the slide, you can see the co-crystallized structure of these 2 compounds, and by, let's say, mere visual inspection of the co-crystallized structure, I have chosen to select this substructure as a pharmacophore constraint in my FTrees farm query, which is also highlighted here as in the 2D structure given by the GUI, and with a similarity constraint of 0.9. And in table, we can see the result that I have obtained by running this similarity search or F3 similarity search. And for the first compound, so BDB219, we can see that at 1%, the enrichment of non-actives with the F3 farm is identical to the one that I have obtained with the regular, let's say, F3s. But at 5%, where F3s dropped to basically a random value of the enrichment factor, which is 1, I have an improvement to 2.5. So it tends to, say, have a higher enrichment at 5%, while this drop at that I have obtained with F3s with 1 at 5% is indicative probably also of the low area under the queue that I have had for this specific case. If you remember, all these cases I had an area under the queue that was below 0.5. So this means that even if there is a high enrichment at I would say early stage of the ranked database, at some point it must drop. In this case, it happened already at 5%, but this was prevented by including this pharmacophoric constraint with F3's farm. And in the second case, so the bottom 2 rows, you can see that once again, the enrichment factor 1% is identical with 2.5, while at 5%, so with the F3s alone, I had 0.5, which is, again, this case is below, let's say, a random selection. And with the F3s farm, I had obtained a value of 1. I wouldn't call this necessarily as a meaningful improvement because 1 is, again, comparable to random. But at least, let's say, we don't have seemingly active deselection active compounds. So as a say, key message for the study case, I would say that for the first query, the modulation of the query by including this pharmacophore constraint has worked in the sense that it was able to prevent this steep drop in enrichment factor at 5% of the ranked database, while in the second case, it was more or less comparable. And in the second case, so for androgen receptor, again, you can see the structure of the co-crystallized compounds in the binding site. And again, I have chosen more or less arbitrarily to select this nitrile substructure as pharmacophoric constraint for the F3 similarity search, and again with the same similarity value of 0.9 as done in the previous study case. So you can see again also how the substructure is highlighted by the query generator or the GUI of F3s. And as a result, so for the first compound, I already have a high enrichment of non-actives at 1% with the F3s alone, and by including the pharmacophoric constraint, this has improved slightly to 10. I say slightly because this is, let's say, a high enrichment with F3s, and I have again a high enrichment with the F3s far. But then I think that the 5% starts to be a little more significant because from 2.5 with the F3s alone, it improves to 4, so it's almost double, let's say, with the F3s farm. And with the second reference compound, again, I'm in this situation where with the F3s alone, I already had a relatively high enrichment of non-actives at 1%, which was 7.5. And with F3s farm, it drops a bit, so to 5. But again, a little bit like in the first case, I would say that it is a high enrichment that more or less stays high with both methods. And at 5% instead, I have a quite significant improvement in terms of enrichment factor. So from 1.5, which is almost close, let's say, to a random selection, to 5 with F3s farm. So basically, it stays stable, the enrichment factor from 1 to 5%, which is, I think, a meaningful improvement in this case. So with respect to, say, to the first study case, I think that it worked quite well for both the reference compounds. Okay, so to summarize the results obtained in this retrospective virtual screening, what I have found is that F3s was able to prioritize in most of the cases non-actives with respect to decoys, and the performance were comparable to those obtained with the ECFP4 and GPI DAF3 in terms of median area under the curve. However, the results obtained with F3s seem to be less sensitive to the specific reference compound chosen, which, as I have discussed before, may be an interesting feature in a virtual screening setting. And finally, I had also run, let's say, some calculation to start getting a feeling of how including pharmacophoric constraints can help to improve the prioritization of non-actives when some information, in my case, it was the observation of the co-crystallized structure is available, so that one can effectively modulate, let's say, the fuzziness of the representation by of the structure representation by including similarity constraint to specific substructures. Okay, so here I switched to the prospective application, and so this virtual screening was run on a quite novel target for which there are no approved drugs, although some active molecules were available. Had a quite rough activity landscape with several activity cliffs, which means that small changes in the 2D structure of these non-bioactives in many cases would correspond to a high difference in biological activity. And the binding site of is basically constituted by a protein-protein interaction surface. And to run this virtual screening, I have selected 2 bioactive compounds as a reference, which are more or less the head of the 2 series that we had at that point in time. And it also to be noted that this application is not a direct follow-up of the previous retrospective virtual screening. In fact, they were carried out, say, in two separate instances, and this target is actually not even included in the dataset used for the retrospective virtual screening. However, we can still have a look at the results obtained here to get more feeling of how FTrees work and compare those to the fingerprints. And so this is the protocol that I have followed to run this virtual screening. I have basically ran two virtual screenings in parallel. One by using 2D fingerprints and Tanimoto similarity. And the 2D fingerprints chosen for this virtual screening were, again, max actual keys and ECFP4. And the third fingerprint was chemical fingerprints included in the Chemaxon suite. And then, of course, F3 similarity. In this case, F3 similarity with the pharmacophoric constraints. I have chosen two constraints that basically were chosen again because a structure was available for these compounds, a 3D structure, so that and also several reactive compounds, so that we could make our mind of which substructure may be more important for activity on this target. So I have chosen these two substructures and set a constraint of similarity of 0.9. In the case of the 2D fingerprints, to come up with a final selection, which the target was to select somewhat around 100 compounds, I have included the top 30 compounds of each ranking obtained with each of the three different 2D fingerprints, and any compound that would rank among the top 300 positions in all three of these rankings in order to find also some consensus. And this led to a selection of 83 unique compounds. And while for F3's farm, so about 13 cancer compounds, or the initial 460,000, were able to, let's say, be selected considering the pharmacophoric constraint that was applied. And among those 13,000, I have picked the 100 that had the highest global F3 similarity. And also, I have mentioned that the database chosen for this virtual screening, it includes about 460,000 compounds, which are the Evotec HTS deck, and we have chosen this database in order to have immediate access to the selected chemical matter. So now the bad news is that unfortunately this selection did not lead to the identification of new active compounds. Yeah, this was somewhat disappointing at first, but then again, the question that this screening was really trying to answer is if in this collection we had some structural analogs in terms of 2D fingerprints or F3s for those two references that I have chosen that would show some activity on our target in order to perhaps be able to expand a little bit the SAR around these compounds. So the answer was no, but we had actually more success later with different methods, but these are actually not covered in this presentation. It is focused on let's say, ligand-based methods focused on F3s in comparison with 2D fingerprints. Okay, so the first check that I wanted to carry out on this selection, conduct with the F3s and 2D similarity, was how chemically diverse those selections were. And I have found that the top 100 compounds so selected with the F3s carried 59 unique scaffolds. So in order to get these scaffolds, I have used RDKit, so calculated this Merkle scaffold. You can see an example in the bottom box where, for example, ATP is shown as a full structure, and also the scaffold, which basically includes only the ring systems and the link between them. And so this can give us an idea of how chemically diverse are this ensemble of compounds. There may be different ways of to assess chemical diversity. I just chosen this for convenience. So in this case, so for FTrees, I have 59 unique compounds, and interestingly, this was the highest value that I have obtained compared to the individual 2D fingerprints. So I went back to take the top 100 for each ranking obtained with the individual 2D fingerprints, so ECFP4, MAX, and chemical fingerprints, and you can see in table the values of the number of unique scaffolds or for which FTrees is the highest. And when I have merged the results obtained with the individual 2D fingerprints in the final selection that was 83 unique compounds, I have obtained 46 unique scaffolds. Now, considering that you have a lower number of compounds, so 83 compared to 100, this is a ratio somewhat comparable to what I have obtained with the FTrees. So I think the key message here is that the F3 section seems to be more diverse compared to individual fingerprints and comparable to what I have obtained by merging three different 2D fingerprints. And the next check that I carried out was how much would those selection overlap. So basically to have a feeling of the redundancy of these methods if they were at all redundant. So in the top 83 compounds, so the selections have obtained by merging the rankings of the three different 2D fingerprints, I have found only 9 compounds that were shared with all the 13,000 compounds which initially passed the pharmacophoric constraint applied with the F3's Pharm. So I think this is really small, so 9 in between 83 and 13,000. So this was, I think, good in the sense that the two selection not redundant and if you want complementary. And when focusing on the top 83 compounds with 2D fingerprint and top 100 with F3's Pharm, only 5 of those were, let's say, still present as a overlap between the two selections. So all in all, I think that these two methods are quite complementary to each other. So to summarize this part of the work, this ligand-based digital screening unfortunately did not lead to the discovery of new active compounds. However, I think it was useful to get a feeling of how F3s work and how does it compare with the 2D fingerprints. And what I have shown, also seen, is that there is a very limited overlap with this within these methods, and that the selection obtained with the F3s was more diverse compared to individual 2D fingerprints. And also, as I mentioned before, we did have more success in this project by applying different methods that included more, say, structure-based approaches, analysis of the experimental structures, modification of non-actives, docking, and so on and so forth. But yeah, this is not covered within this presentation. Okay, I would like to thank Iñaki Morao, who's a principal scientist here at Evotech, who helped me to carry out this work and somewhat kickstarted, let's say, my adventure into applying F3s. And please, if you have any questions or answers, and I will try to give some answers.